Okay, so we got a talk from three folks from Anshul's lab. So they're going to introduce themselves since there are three of them, but this is going to be telling us about how to evaluate regulatory genomics models. So thank you guys.
Hi, we're Austin, Aman, and Arpita, and we are presenting DART-Eval, which is a set of DNA language model evaluations that focus on regulatory DNA. Yeah, so self-supervised models, they've proven extremely useful at learning complex and contextual relationships, first for text, and also more recently for proteins as widely adopted protein language models. And this begs the question, can this approach also be applied to genomics? Perhaps a DNA language model or a DNA LM? Well, despite how recent the concept is, a lot of DNA LMs have come out. And in our study, we specifically look at whole genome DNA language models that try to capture a universal representation of all genomic information by pre-training on all of the DNA from one or more species. However, learning this universal representation is actually very challenging. So far, most of our success has been in protein coding regions, which are relatively dense and homogeneous and encode a coherent biological function, which is protein structure. However, these regions form only a small fraction of the overall genome, which contains several different types of functional elements, each representing distinct biological processes operating under a completely different set of rules. And this is further complicated by the predominance of neutral DNA, whose sequence is not relevant to biological function. And what's worse, all of these different types of elements are interdispersed as a big messy soup. But for DART-Eval, we don't really attempt to evaluate every type of functional element, but rather we focus specifically on non-coding regulatory elements, which encode the programs controlling gene activity. And a hallmark of non-coding regulatory DNA is its cell state specificity, where the same stretch of DNA can be read in very different ways depending on cell type due to the presence of different transcription factors and cofactors. And this context specificity makes regulatory DNA amazingly expressive, but it also poses a unique challenge to any attempt to model it. But unfortunately, the rate of model development has greatly outpaced the availability of robust evaluations, especially for regulatory DNA. First, there aren't that many benchmarks that examine context-specific regulatory activity, and moreover, they often only involve surrogate predictive tasks that don't address key downstream applications. For example, oftentimes we see that a DNA LM is fine-tuned to classify the type of regulatory element and solely evaluate it on the same task. But this misses the practical application, which is not to classify elements, but rather to reveal sequence motifs or design synthetic sequences. And lastly, they suffer from dataset design issues, such as the lack of compositionally controlled negatives or not accounting for LD when scoring variants. And to fill this gap, we introduced DART-Eval, which is an adaptable benchmarking framework consisting of a series of tasks that assess a wide range of regulatory DNA use cases for DNA language models.
All right, so now we'll go into some more detail about our modeling approach. And we apply these evaluations on 4 prominent and recently published DNA language models. And these are DNA BERT2, Jenna LM, Hyena DNA, and nucleotide transformer, who you just heard about in the last talk. And then for each task, we have some combination of zero-shot and supervised evaluations, along with rigorous baseline models. And so in the zero-shot regime, we look at the last layer embeddings of the models, along with their predicted sequence likelihoods for the sequences in question. And in the supervised setting, we do probing, which in our case entails freezing the pre-trained model and then training a small CNN on the last layer embeddings. And we also do fine-tuning using LoRa. And LoRa is a parameter-efficient fine-tuning method intended for large models where fine-tuning the entire model might be infeasible. And then for each task, we also have baseline models that are ab initio, or trained from scratch. And these are typically dilated convolutional architectures. So now let's go into each of the tasks and the results. So starting with our first task, which again is the easiest, it involves testing whether the models can detect any kind of regulatory DNA. And on a high level, this task involves comparing regulatory sequences to controls and determining whether the models can tell those apart in zero-shot and supervised settings. So going into this in more detail, the positive examples in our dataset for this task consist of approximately 2.5 million candidate cis regulatory regions, CCREs, as defined by the ENCODE consortium. And these are basically high-confidence regulatory regions. And our negative set consists of these sequences, but shuffled in a way such that dinucleotide frequencies are preserved. And this kind of shuffling means that the positive and negative sequences are compositionally equivalent and are not affected by confounders such as GC content or differences in dinucleotide frequencies, which has been a big problem with previous benchmarks. And the only difference then between the two categories of sequences is the presence of regulatory motifs, which is what we expect the models to detect. So we test this in two ways. In the zero-shot setting, we look at model likelihoods, and we expect the models to assign a higher likelihood to the regulatory sequences than to their negatives. And in the supervised setting, we train classifiers to differentiate the two classes. So first of all, our ab initio CNN baseline proves pretty effective at discriminating the two categories. And then if we look at the zero-shot setting first, we see that DNA BERT2, for example, is pretty competitive with the baseline, albeit a little inferior. And all the other models generally show competitive performance as well. And then when we get into the supervised settings, the DNA LMs even start outperforming the ab initio models. So overall, what we're seeing is strong accuracies across the board for this task. And I would argue that these results generally reflect the lack of difficulty of this task. And we see this one as kind of the preliminary bar that we have to clear before getting into the more complex evaluations. So speaking of which, our next task is intended to see if the models can graduate from recognizing any type of regulatory DNA to actually recognizing specific regulatory motifs. And we do this in a zero-shot setting with testing both likelihoods and last layer embeddings. So in more detail, we use a list of 1,443 transcription factor binding motifs as produced by the Hokomoko database. And we also have 100 of the shuffled background sequences from the previous task. And then for our positive set, what we do is we insert each of the motifs into each one of the background sequences. And then for the negative set, we do the same thing, except with shuffled versions of these motifs. So let's take a look at DNA BERT again, specifically in the likelihood setting. In here, we define an accurate prediction as one in which the model assigns a higher likelihood to insertion of the true motif than to the shuffled one. And then we see that model performance is generally effective, but not perfect. So the plot here shows the CDF for the accuracy of each motif, and the line for convenience is at 70% accuracy. And we see that around 20% of motifs exceed that threshold. So the model seems to identify a good number of motifs pretty well, but not all, and there is a large number for which the accuracy is pretty poor. And this trend carries over to the other models as well. Some are better than others, but all of them show pretty reasonable performance for a large number of motifs, with poor performance on some of them. And the numbers again show the proportion of motifs with over 70% accuracy. However, the last layer embeddings, on the other hand, seem to show very little information, if at all. So here, we define an accurate prediction as one in which the embedding distance from the neutral background to the positive is greater than the distance from the neutral to the negative. And all accuracies seem to be around 50%, which essentially indicates random predictions and is not really informative at all. So overall on this task, we see very mixed results.
Moving on from identifying specific transcription factor motifs, it is also important to understand cell type specificity. As we said earlier, a hallmark of non-coding regulatory DNA is its cell state specificity. So it's important to evaluate whether models can truly learn cell type specific regulatory vocabulary. To achieve this task, we use chromatin accessibility peaks measured by a TAC-seq for 5 cell types that come from very different regions of the body and have differential accessibility patterns. Moreover, each of these sets of differentially accessible peaks contain distinct regulatory DNA, including specific motifs that characterize them. We first evaluate models using zero-shot embeddings, where the model embeddings are produced on these differentially accessible peaks in each cell type. The baseline embeddings are produced by featurizing each peak as a vector of motif counts, and then these embeddings are clustered. We evaluate the embeddings in 2 ways, visually and quantitatively. Visually, we look at the UMAP of the embeddings of these sequences, and quantitatively, we perform k-means clustering and use mutual information to evaluate the clustering quality with respect to the true labels. Here, we can see that the bag of motifs baseline approach is able to identify modules that can separate by cell type. On the other hand, let's take a look at DNA BERT2. The model embedding clusters are not separable at all by cell type compared to the embeddings. And the mutual information scores further support what we see, with the DNA BERT embeddings having a much lower score compared to the baseline embeddings. The other DNA language models follow suit and are unable to classify cell types similarly to DNA BERT2 and perform much worse than the baseline both visually and quantitatively. So the models did not perform on par with the baseline in zero-shot space, but what if we help the models using supervision? This time, we will train a 5-way classifier on chromatin accessibility peak sequences coming from each of the 5 cell types. We measure the performance of the classifier using AUROC, and as we can see here, the ab initio baseline is able to classify cell types well with an AUROC of little more than 0.9. So what about the DNA LMs? Probe DNA BERT2 performs at a much lower AUROC than the ab initio, and fine-tuning we see improves accuracy but does not beat the ab initio. And similar to DNA BERT2, other supervised DNA LMs also perform worse than the ab initio at classifying cell types. And this is across all cell types. So overall, for this task, we see that most models barely perform on par with the supervised baselines, even after these much larger models are fine-tuned. So moving on, we want to evaluate whether models can actually quantitatively predict regulatory activity, which is an even more difficult task. Chromatin accessibility is a strong quantitative indicator of a sequence element's regulatory activity. If a model can successfully provide a quantitative measure of accessibility from sequence, the model can be used to provide a more nuanced understanding of non-coding cell type-specific regulatory syntax. This is a common classical task for which many supervised models already exist, but can we do better with DNA LMs? We evaluate for this using chromatin accessibility peaks from the same 5 cell lines as the previous tasks, along with their GC-matched inactive sequences. We benchmark these models using supervision, where we train models on the peak sets and corresponding negatives to predict the log DNase read counts over the input regions. And we evaluate performance using Spearman correlation between the predicted and true peak counts. The supervised sequence to activity ab initio model is represented by the dashed line here. The sequence to activity DNA BERT2 underperforms the ab initio in both probed and fine-tuned settings at quantitative regulatory activity prediction. And so do the other sequence to activity DNA LM-based models with both probed and fine-tuned performance well below the ab initio. And this happens again across all cell types for all DNA LMs. So most evaluations of models end at this surrogate task, but we cannot just rely on observational performance when evaluating models. It is necessary to evaluate models on true counterfactual tasks, such as our next one. For task 5, we benchmark models on this counterfactual task where the models are evaluated on sequence they have never seen before in their training set. This task tests whether models can predict effects of genetic variants in regulatory activity. This is the most difficult and biologically meaningful task in our benchmark. If models can accurately predict variant effects, we can determine how a single allele is influencing the regulatory readout. To test our models, we use a dataset of chromatin QTL variants present in African and European LCLs and utilize our sequence to activity models from the previous task trained specifically on the GM12878 LCL regulatory activity and not any variant datasets. To benchmark these models, we use rigorously curated variant sets. The causal QTLs are defined as those that are statistically significant and located within peak regions, thus drastically reducing the confounding effects of linkage disequilibrium. The control set consists of statistically insignificant QTLs that also fall within peaks. We first evaluate models in the zero-shot setting, where we measure the cosine distance between the embeddings of the reference and alternate alleles, with the causal variance expected to produce a higher embedding distance. So our ab initio model, again, is a sequence to activity baseline model from the previous quantitative prediction task. And it's important to note that it was not trained on any variants. We measure performance using AUROC, and as we can see here, the baseline performs quite strongly on both data sets. DNA BERT2 zero-shot AURC is well below ab initio at about 0.5, and other models perform similarly across both data sets. So next, we try to help the DNA LMs out by using the sequence to activity DNA LMs trained on the GM12878 LCL chromatin activity from the previous task to predict variant effects, again, not trained on any variants, and we compute the absolute predicted accessibility difference between the two variant alleles and use that as our metric. Unfortunately, supervision on chromatin accessibility does not help DNA LM performance either, and we can see that the DNA LMs display significantly inferior performance to the ab initio models across all settings. So this task demonstrates the necessity of real counterfactual tasks to determine true performance of the models.
So to recap, we evaluate state-of-the-art DNA language models in zero-shot, probed, and fine-tuned settings across increasingly difficult regulatory DNA evaluations. And we find that although DNA LMs excel at simpler tasks, their performance degrades with increasing complexity relative to more resource-efficient ab initio baseline models of regulatory DNA. And all of these analyses we package into our adaptable DNA-LM benchmarking framework, which provides 5 families of biologically motivated tasks and datasets that evaluate diverse aspects of regulatory DNA. And that being said, there is a whole world of other aspects of regulatory activity that we have not touched. These include long context effects, which we can infer from histone ChIP-seq or Hi-C. It also comparisons of regulation across species. And lastly, other types of functional elements such as UTRs and splice sites. And finally, it is clear from our experiments that DNA-LMs have to get better before being practically useful. So we argue that the unreliable performance comes from the noisy and imbalanced whole genome pre-training data. So the lowest hanging fruit, even before optimizing for architecture, is to improve data curation and data training. And these can include training local short context models on specific classes of functional elements, such as promoters, 3' UTRs, and coding sequences, and adaptive sampling of elements to account for class imbalance, and explicitly conditioning on cell state context. But note that all of these strategies depend on some sort of annotation beyond just a whole genome sequence. And it is an open question whether universal DNA LMs can be built without the help of these annotations. Yeah, so thank you so much.
Do we have time for a few questions?
Hi, thanks for that great talk. I had a question on the choice of baseline. There was a paper here last year doing some benchmarking on regulatory elements, and it showed that non-neural models, so like support vector regressions or other types, can be competitive or even outperform things like DeepC or ab initio models. Do you have any further plans for additional baselining or any thoughts on ab initio models there?
Yeah, that's a great question. I think there are a lot of potential baseline models that can be used, but I think one important thing to note is that a lot of the models that have been used for baseline benchmarks in the past, like DeepC, for example, are trained on data that is not quite at the state of the art, because now we have really rich quantitative data for a lot of these regulatory readouts, and so I think the most competitive baselines are models that are directly trained on that. So some of the models that even have been published a few years ago now are not quite at the forefront, and it's important to make sure that we use supervised models trained on the most recent data. So I think similar to the dilated convolutional architectures we have here, and I would wager, we can totally try, but I would wager that those would beat more SVM type models on this type of quantitative data.
I had a question for you guys that all the DNA language models, when they're pre-trained, they use some folds to train and test. But when they're compared with all other sequence expression models, for the downstream tasks, they don't use the same folds as the pre-training. So do you think a lot of their comparison could be leaked completely and they should be retrained using the same folds as the sequence to expression models?
Yeah, that is a great question. We definitely agree with that. For our purposes, we don't bother retraining all of the pre-trained models on a rigorous howled-out test data set, which does give them a little boost. So there is a possibility that the DNA language models here have the additional help of some amount of data leakage from the training set to the from the pre-training set to the test set. And we believe that this is probably a bigger issue for the easier tasks, like the CCRE classification, and probably less of an issue for the more of the counterfactual tasks. Yeah, and I guess in fact, for the variant prediction task, that is the only task where we guarantee to not have data leakage because none of the variants are present in the reference genome. Yeah, thank you.
Thank Aman, Arpita, and Austin again.